Welcome guys to 2010 Biology Practical Marking. It is in the evening and I want to present it to you. It's evening in Kenya, so I will present it and I hope you are going to enjoy as you learn the tricks of the way practicals are marked. So, welcome. KCSE Biology Practical of 2010. Have you ever wondered what sets apart an exceptional biology exam paper from the rest? What if you could peek behind the curtain and see exactly how the KCSE Biology Practical of 2010 was marked? In this exclusive video, we are unlocking the secrets of marking schemes that have shaped countless students' future. Join us as our esteemed teacher and senior examiner, renowned for their in-depth knowledge and years of experience, takes you through the meticulous process of evaluating biology practical exams. By the end of this presentation, you not only understand the critical components that examiners look for, but also gain insights that could drastically improve your exam performance. Whether you are a student aiming for top marks, a teacher seeking effective strategies, or just someone curious about the inner workings of exam assessment, this video will bridge the gap between confusion and clarity. You will discover how precise marking criteria can transform your approach to practical exams and help you excel in your academic journey. Don't miss this opportunity to learn from the best and enhance your exam skills like never before. You are all welcome. So, you are provided, this is question 1, you are provided with a Visking tubing, a solution labeled L, iodine solution labeled solution E, Benedict solution labeled solution F, and a piece of thread. Tie one end of the Visking tubing tightly using the thread provided. With the help of a syringe, put 10 ml of the solution labeled L into the Visking tubing. Tie the end of the Visking tubing tightly. Ensure that there is no leakage at both ends of the Visking tubing. Wash the outside of the Visking tubing with water. Place the Visking tubing upright in a 100 ml beaker. Add distilled water into the beaker to reach the level of the liquid in the Visking tubing. Allow the setup to stand for 30 minutes or more. Using 2 ml in a test tube in each case, test for the food substance in the liquid outside the Visking tubing using, you get 6 marks. So here you are going to use iodine solution, which is labeled E, and Benedict's solution, which is labeled F. So there you have the procedure, observation, and conclusion. So guys, I started with the way the question had been set. B, using 2 μ in a test tube in each case, test for the food substance in the contents of the Visking tubing using, you get 2 marks, iodine solution, Benedict's solution, E and F. Notice in these tables, we are not having food substance, we are having test. So this is different from the other. So I'm sorry, the camera just went off, and the phone has told me the lens was dirty, so I wiped the lens. So I'd already taken you through here, but there is no harm of repeating. Using 2 μ in a test tube in each case, test for the food substance in the contents of the Visking tubing using, you get 2 marks. Iodine solution is solution E, Benedict's solution is solution F. So you have test, procedure, observations, and conclusion. You find now here, instead of writing there food substance, we have written test. So the test is known as iodine solution test. Okay? And this one is known as Benedict's test. So iodine test and Benedict's test. But if it was food substance, now you'd put the food substances. But you should also still be knowing the reagents. So in C, you are accounting for your results in A and B above. In question 2, the photographs labeled JKM1, M2 are section of mammalian heart. Examine them. So this is where I was telling you in the confidentials that these things were in the question paper. So photograph J, photograph K, photograph M1, and photograph M2. The blue, green, and cream strings go through various blood vessels and end up at various chambers of the heart. For each string, name the chamber where the string ends and the blood vessel through which the string goes. 8 marks. So string, chamber, blood vessel. So we have blue there, green, cream 1, cream 2, and then you fill the spaces. Name the parts labeled 3 in photograph K. Then the parts labeled 4 and 5 are walls of 2 chambers of the heart. Account for the differences in the thickness of the walls, 1 mark. Photograph M1 shows 2 blood vessels labeled X and Y, while M2 shows transverse section of the same blood vessels. With a reason, identify the type of each of the blood vessels. 4 marks. X, reason. Y, reason. In photograph K, indicate by letter B the part of the heart which would be cut to expose the bicuspid valve. 1 mark. Question 3, you are having photographs again. For question 3, there are photographs and you can see them. Let me adjust a bit, slightly. So going back, the camera is taking itself back. So you have photograph. The photographs labeled Q, R, S, and T are sections of some parts of plants. Photograph Q, photograph R, photograph S, and photograph T. Photograph T as labels 6, 7, 8, 9, 10. So question 3a, name the type of presentation in the specimen shown in photograph Q, R, and S. You get 3 marks. B, label a seed in photograph R and S, 2 marks. C, name the parts labeled 6, 7, 8, 9, and 10 in photograph T, 5 marks. Giving a reason in each case, name the mode of dispersal of each of the specimen in the photographs Q and T, 4 marks. So Q, reason, T, reason. So let's come now to the expertise, how the examiners looked at those questions. So in question 1A, where you are using 2 needles in a test tube, in each case, to test for the food substance in the liquid outside the Visking tubing, using the reagents given. Test was iodine, where you add a few drops of iodine solution in the procedure. No color change. And then you see starch pre-absent. There are alternatives there which you can, and as I've taught you in the other papers, that you pick only one. So add a few drops of solution E to liquid in the beaker, or distilled water, and the color will change to brown, yellow, orange color of iodine retained. You see starch absent. Benedict's test to 2 ml of the liquid from the beaker and 2 ml of Benedict solution. Solution F is added. The mixture is heated in water bath. The solution acquires a yellow, orange, brown, red, or brown or reddish brown color. All right, reducing sugar is present. If the series of colors, the sequence must be correct. Because here the solution acquires yellow. You know, it's supposed to be green, yellow, orange, the sequence. No, actually you start from blue, green, yellow, orange. Reject red. It is a reddish brown color. So reducing sugar is present, but reject if somebody says simple sugar, all right, or monosaccharide, or specific simple sugars, or little or traces of simple sugar. Why do you ignore simple sugar? There are other sugars which are simple, but they are not reducing sugars. Question 1. Accept, so this is the way it was looked at, accept Benedict's or Benedict's. Reject Benedict if there is no S, but proceed to mark observations and conclusions after putting an omission. This one, omission is inverted V. You just put it where the person was supposed to put it. Wrong spelling in conclusion, you reject. Note, volumes are qualitative tests which show the results irrespective of quantity. Orange color is visible depending on the level of dilution. It lies between yellow and brown. I want procedure for iodine test only once in A and B. I want procedure, observations, and conclusions from Benedict's test only once in A and B. Note that. So we are in B, and you can see equivalence. So using 2 μ's in a test tube in each case, test for the food substance in liquid outside the Visking tube using 2 μ. So using iodine solution and Benedict solution, 2 marks. So procedure is just the same like A, and therefore we don't give a mark there. But observations, solution acquires a blue-black color, so starch is present. Allow blue, black, bluish, black, or dark blue. Reject blue or black, or blue stroke black, or black blue. If anybody says those colors, you reject. And so you don't get the conclusion. Benedict's procedure was repeated in A, so you see there no mark, except small letter B for Benedict's. The solution acquires yellow. If a series of colors, the sequence must be correct. Reducing sugar is present. One small rejected simple sugar, monosaccharide, or specific sugars, or little or traces of sugar. Note, sugar molecules moved, starch molecules not moving. Check from the table to award semi-permeability. Account for the results A and B above, 3 marks. A, Visking tubing is semi-permeable, selectively permeable, allowing small reducing sugar molecules to move out, pass, diffuse out, and not allowing starch molecules to move out. Account for results in B. Here you don't give a mark. Those are just equivalents because there are only 3 marks. I want semi-permeability, selective permeability, reducing sugars moving out, and the starch molecules not moving out to get your 3 marks. Stop marking accounting if conclusions in A and B are not correct. Check for tick or X in conclusions to proceed to account. So you check whether there was a tick or an X in the conclusion so that you proceed to mark the account. Otherwise, if there is an X, you don't proceed. You proceed only if there is a tick. If conclusion is traces or little amount, underline and proceed to mark accounting. C can only be awarded 3 marks if A and B conclusions are correct. If starch, only correct only 1 mark for C. If reducing, only correct only 1 mark for C. If reducing only, correct only 1 mark for C. Semi-permeability mark when starch and sugar, conclusion is correct. Question 2. The photographs J, K, M1 and M2 are section of mammalian heart. Examine them. Photograph J as rebels 1 and 2. Photograph K as 3, 4, and 5. Photograph M1 and photograph M2 as X and Y rebels. The blue, green, and cream strings go through various blood vessels and end up at various chambers of the heart. For each string, name the chamber where the string ends and the blood vessel through which the string goes. All that will give you 8 marks. So, string, chamber, blood vessel, blue goes to the right ventricle chamber, and then blood vessel is pulmonary artery. Green, left ventricle, blood vessel, branches of aorta. Cream, right auricle or atrium, then vena cava. Cream 2, left auricle, atrium, pulmonary vein. No tying, mark independently. Reject plural. We are still in question 2. Name the part labeled 3 in photograph K. That one is known as septum, or you can call it interventricular septum. The parts labeled 4 and 5 are walls of 2 chambers in the heart. Account for the differences in the thickness of the walls. So, 4 is thicker than 5 because the latter pumps blood to the lungs and 4 forms the wall of the chamber that pumps blood to all other parts of the body of a long distance. So 4 is wider, or 4 is thicker or wider or more muscular. Like you can see, 4 is more muscular than 5 because 5 forms wall of the chamber that pumps blood to the lungs, that pumps blood a shorter distance, and 4 forms the walls of the chamber that pumps blood for a longer distance. So that's the way you are supposed not to put strokes, as I told you the other day. Photograph M1 shows 2 blood vessels labeled X and Y, while M2 shows transverse sections of the same blood vessels. With a reason, identify the type of each blood vessel. 4 marks. X is vein, accept veins, because there are 2. Reason, it has thin walls or it has thinner, or it has less muscular walls. Why is artery? Accept arteries. Reason it has thick muscular walls, or thicker or more muscular. Reject wide rumen or narrow rumen because they have the same diameter. So you cannot use the rumen there. If you measure the diameters of those photographs, they are the same. In photograph K, indicate by letter B the part of the heart which would be cut to expose the bicuspid valve. The tendons direct you where the bicuspid valve is. All right. So you can see where the B is labeled. Okay. You can allow the arrowed label, okay, for B. All right. 3, 4, 5 are still there. Photograph K. Reject if labeled bicuspid valve and not B. Allow if labeled area where bicuspid valve is found. Question 3, our last question. The photographs labeled Q, R, S, and T are sections of some parts of the plants. Photograph Q, photograph R, photograph S, T as labels 6, 7, 8, 9, and 10. Name the types of presentation in the specimen shown in the photograph Q, R, and S. Q is marginal, R is axial or central, S is central. Reject free central or axial spelling. Label a seed in photograph R and S. So you can see where we have labeled the seeds. They are very visible. All right. Okay. Transverse section for that one, the photograph on the left, they are saying it's not clear. It does not have clear oculi for free central presentation. And longitudinal section is free central, and transverse section is central. Name the parts labeled 6, 7, 8, 9, and 10 in photograph T. 6, epicup, or you can call it the exocup. 7, seed, accept cotyledon, or seed of mango, or seed of avocado. 8, endocup, reject endocup filled with a fluid. Mesocup, reject fibrous mesocup. 10, remains of flower stalk or fruit stalk or pedicel. Reject stalk alone. Giving a reason in each case, name the mode of dispersal of each of the specimen in photograph Q and T. Self, or you can say for Q, self, or you can say self-dispersal, or you can say self-dispersed, or you can say self-explosive, or self-explosion, or self-mechanism, or self-explosive mechanism. In fact, the best one is self-explosive mechanism, but all the others are correct, except explosive mechanism without self, or explosion mechanism. Reject explosive alone, or explosion alone, or self-dispersion. Reason, it has such as, or lines of weakness, or lines of dehiscence along which it spreads. All right. T is animal, or animals, or animal dispersal. Reject the specific names of animals, e.g. man, or band, or insect. Reason, fleshy, succulent, fleshy mesocarp, brightly colored mesocarp. Reject juicy. So, having come to the end of the question 3, now I just give an acknowledgement saying that this work is truly a team effort. There are too many who have inspired me to come out with this video. I thank everybody, including my family members, especially when typing this 2010 paper. I slept at around 2:30 p.m. I'm also grateful to all my teammates during marking exercise of paper 3, who in one way or the other participated in coming up with the final draft of the marking scheme during our meeting of senior examiners. But I also give special thanks to ChatGPT, which is an AI language model developed by OpenAI for providing assistance in creating content for this video. Like giving the hook, curiosity hook, and the bridging up. Otherwise, most of the content is from me because I participated in the marking. So guys, that comes to the end of the way Biology Practical of 2010 was marked. I welcome you to comment. If you find that the way I've presented has got any weakness, please suggest so that I improve. Recommend these videos to your friends, and God will bless you. All right. So, may I see you in my next video, 2011. Remember, as we go on, we are minimizing correction of materials, live materials, and we tend to be using now photographs. And because technology now is advanced, photographs will look exactly like the real specimen. So guys, goodbye and good night, wherever you are. If you are in Kenya, it's good night. If it is in the other places, it's good morning or good afternoon. So, bye, see you next time.